Okay. Again, thank you so much for the organizers to have the opportunity to be here. I'm actually really happy that I can follow the previous talk because a lot of background, you guys are now all set for that, and I don't really have to talk about that, and hopefully everyone can follow so much easier. Just to give you a little bit of an idea, so my name is Markus, and I work at the Rare Disease Translational Center up at the Jackson Laboratory in Maine. And to give you sort of a very general idea for our group tries to develop model systems that ultimately allow the testing of therapeutic strategies with the idea to provide preclinical data for IND applications. And our process usually starts in a very similar fashion across different projects. It usually starts with identifying patient-specific variants. Together with our partners, we try to then anticipate potential therapeutic strategies. And then we combine these two pieces of information with the idea to develop a model system. We are a mouse institute, so a lot of our focus is developing mouse models, but equally acknowledging that an animal model is not always a faithful system to study. We also develop, as we just heard, human cell lines that may enable better therapeutic development if a mouse isn't working perfectly. And then ultimately, we use these data to go into preclinical data generation. And this approach can be technically applied to a large number of rare genetic disease. I particularly focus on rare neurological disease, including KIF1 alpha. And there are two parts that I want to touch on. On the one hand, the model generation, and then on the other hand, our efforts for therapeutic testing. If we sort of take a look at the model system that are available for KIF1 alpha, we have learned over the last years that cell-based systems or systems like C. elegans have actually been incredibly useful as a model system. Mice, unfortunately, have been rather limited, for the lack of a better term. Although one exception might have been a mouse model that we refer to as the lag dragger mouse model. It's a spontaneous mutation that occurred at our institute. It has a pathogenic variant in the motor domain of KIF1 alpha. It's a variant that has not been observed in the patient population, but the mice display a phenotype that is very reminiscent of what one might see in the human population. So for example, these animals develop very severe hind limb paralysis, usually around 4 weeks of age. This is usually associated with peripheral neuropathy, which we similarly observe in the patient population. There is Purkinje cell degeneration, which kind of goes along with the idea of cerebellar atrophy, but you also have survival defects. These animals normally don't survive past 8 weeks of age. And aligning KIF1 alpha with other neurological conditions, these mice also have an increase in serum NFL, which is a neurofilament light chain protein, and it is used as a blood biomarker, as a non-invasive blood biomarker across a large number of neurological conditions, both in mice as well as in humans, to track disease progression as well as therapeutic outcome. However, as I mentioned, the lag dragger mutation is not a variant that has been observed in patients, so we decided to generate mouse models that carry patient-specific variants with the idea that we can hopefully develop a model that would allow us to perform preclinical studies with those mouse models. When we introduce these patient mutations in a homozygosity, meaning both copies are affected, this is actually not compatible with life in an animal model. These animals die shortly after birth. And with that in mind, we focus much of our studies trying to characterize mice that have one healthy copy and one affected copy, which is more similar to that of a patient. When we think about characterizing these mice, we can look at a lot of different aspects. So in this case, we take information that we know from the patient population as well as from mouse work, meaning we can look at motor and nerve function in these mice, and we can do this through a large battery of assays, which can include rod, treadmill. We can look at nerve physiology, which we call CMAP, and we can also look at grip strength, and we can use non-invasive biomarkers like NFL. And we can do this in a longitudinal fashion to understand if there are defects that are developing over time. We can also take a look at the brain more specifically. For instance, we can look at the memory function of these mice. In mice, we do this with a test that's called contextual fear conditioning. We can also place electrodes into the brain, very similar to an EEG, to kind of get an idea about neuronal activity. And we can take an even closer lab into the histology of those brains or even the spinal cord. All that being said, while we did a large number of behavioral assays, actually the majority of those assays didn't result in any noticeable, overt, significant defects, perhaps with one exception, which are Purkinje cells that actually do show a very noticeable defect. To sort of just give you an idea about that, if you look on the left-hand side, you can see the cerebellum. The cerebellum in mice as well as in humans is placed in the back of our head. It's important for motor function, motor coordination, and balance. And if you take a look on the right-hand side towards the blue bar, you're looking at the quantification of Purkinje cells, which are drastically reduced in P305L mice. But as I mentioned earlier, while we have mouse models, you can clearly see that these animal models are not necessarily always giving us what we really wanted to look for in an animal model. And even though we still study those mice, particularly the mice for the T99M mutation, we do develop other models. And I will make it very brief because the previous talk actually touched on that very nicely. We develop human iPSC lines with the idea that you can have a human model system to then use that for therapeutic development. And in this case, we introduced 7 different mutations for KIF1 alpha in those cells. And as we have just learned, iPSC cells are a great system. You can differentiate them into your cell type of interest. For us, as neuroscientists, neurons are incredibly important. We can differentiate them into motor neurons, or as we just learned, also in cortical neurons. And you can use these cells to screen and validate therapeutic strategies, as well as studying the biology. And just to give you a sense, because we have seen pretty pictures, and I wanted to throw in pretty pictures as well, we can look at the differentiation of those iPSCs into neurons over time. So on the left-hand side, you have these cells, and we can look for markers that are indicative of these cells being progenitors, like ISL1. Over time, when we culture them correctly, we can see markers of early motor neuron nature, like HB9, and even further, the motor neurons will mature into mature motor neurons. Sort of taking a step from the model system, either mouse or human, whatever is applicable in the best way possible, we also try to anticipate therapeutic approaches and use the best model to test those. And with that, I want to focus for the second part really on therapeutic approaches. And the part that I want to focus on today is gene editing, with the idea that you can actually correct a pathogenic variant and permanently correct it for the rest of life. However, I do want to acknowledge that most therapeutic strategy development often relies on simultaneously exploring other opportunities, simply with the idea we don't know which one will work the best. And we have heard about this. My mouth is getting really dry. Sorry. We have heard about this for the antisense oligonucleotide development. We have heard from Anne Loram, which is a fantastic approach to develop ASOs. We also know that conceptually one can explore the idea of gene therapy in KIF1 alpha. In mice, we do that. Thank you so much. In mice, we can do this by overexpressing human KIF1 alpha, and these are still studies that are ongoing. In addition, I want to mention that there are also more traditional ways of trying to modulate disease by piggybacking existing neuronal pathways that are considered to be important for neuronal health, and we do that in collaboration with other researchers. But as I mentioned, I wanted to talk about gene editing. And it is really great that we had the presentation from Anne Loram just a few minutes ago, because as we have learned, it is not just important developing a therapeutic, but ultimately many of us are realizing we actually need a pathway to go from an experiment into the clinic. And ASO development relies on many years of experience in which an actual administrative process is in place that can be piggybacked. In the context of technologies like gene editing, we actually don't have any path in place. And a few years ago, a large number of investigators, together with regulatory advisors and the NIH, came together and decided to create the Somatic Cell Genome Editing Consortium with the idea to actually put a pathway in place for gene editing to be no longer an experiment, but something that can actually go into the clinic. This consortium has a large number of groups involved that are really working on different aspects. The members that we are working with includes David Liu, who really pioneered gene editing. It includes Amanda, who is living and breathing gene editing as well, and Stephen Gray, who has long-standing experience in gene therapy, viral delivery, and the transition from preclinical work to clinical work. And for the partnership with these people, we apply gene editing in the context of rare neurological disorders, with the goal that all of our preclinical studies go actually into clinical trial over the course of like 3 to 4 years. We selected a number of projects to be part of this consortium, which includes spinal muscular atrophy, Friedreich ataxia, Huntington, and Rett syndrome. And while this is our primary goal, the goal for the consortium always has been to develop a path that allows other rare genetic disease to follow the exact same pipeline. And with that in mind, we have expanded our work together with David Liu towards other neurological conditions, including alternating hemiplegia of childhood, which we just recently published about, Zellweger disease, but we also included KIF1 alpha. And with that, I sort of want to take a little bit more time about the gene editing approach for KIF1 alpha. If we think about gene editing, it can usually be categorized in 2 components, either base editing or prime editing. We use prime editing because it is more amenable for our experiments. And prime editing is often referred to as a search and replace function, meaning if you have the 2 components that are critical for prime editing together, they can find in the genome the pathogenic variant and can then actually rewrite the DNA to correct the DNA permanently. The components that you need for that is a Cas9 enzyme that is modified to have a reverse transcriptase function, as well as the guide RNA. And when we think about developing gene editors, there's usually a multi-step process involved. And this usually requires screening through a large number of gene editor combinations to find an editor that can actually effectively correct the DNA. In many cases, this is being done at first in immortalized cells. Those cells are very easily available. They're very easily to grow. But these cells do require the introduction of the pathogenic variant, which takes time. By the time you have identified some sort of lead editor, it usually goes into another cell type, which is a primary cell type that is either derived from a patient or from a mouse, depending on the goal of your study, to validate that the editor can work. And then from there, we usually move into animal work, where we confirm that the editor can actually work in a living organism. And then from there, we generate preclinical proof of concept data that an editor can actually correct disease-related phenotypes. And Angus, who is here in the audience, who works in the lab of David Liu, was asking the question whether or not we can actually skip the very first step with the idea to accelerate this process. And therefore, he decided to use computational tools together with our primary cells that we can isolate from our leg dragger models, and asking, can we actually identify in a rather rapid fashion gene editors? And without undermining the work that he has been doing, I just give a very small snapshot. And what you can see on this slide is a different number of prime editors that are combined with different guide RNAs. These are the 2 components that are required in order to get gene editing to work. And there are 2 readouts that we are focusing on. On the one hand, you have the green bar. You want the green bar to go as high as possible. This is the level of how much we can correct the mutation. And on the other hand, you have the red bar, which is how often does the editor make a mistake. And we want the mistake to be as low as possible. There are 2 editors that sort of have a little bit of a reddish background, which is sort of indicating 2 of the leads that we wanted to validate in vivo. When we think about gene editing in vivo, particularly in the context of the nervous system, currently the best way to deliver something into the nervous system is through a harmless viral delivery. And I'm using harmless a little bit with quotation. It is, in the end of the day, still a virus. But in this case, we deliver the virus into mice, and we deliver them usually as early as possible, which is directly after birth, and we deliver them directly into the brain. And then we let the animals live for a couple of weeks, making sure that the editor is not toxic to any of these animals, and then equally collecting the tissue to actually confirm that editing does occur in vivo. And here you have a graph that looks very similar to the slide that you have seen before. You have at the bottom our 2 lead editors, and you have these green bars where we try to see level of correction for our lag dragger mutation. And what you can see is that we have very high level of editing in vivo. And more importantly, when you look at the red bars, we have very low levels of making a mistake, which is about less than 1%. We have learned throughout the talks that while we focus on the gene that has a mutation, the gene is usually producing a product which is actually causing the problem in our cells. And with that in mind, we can also look at the editing correction for the product that is being derived from that gene. And very similar, we can see high level of correction for the product that comes from the KIF1 alpha gene. Taking these pieces of information together, Angus's question of can you use primary cells together with computational tools to effectively screen for editors, we do feel very comfortable about using primary cells to speed up the process. We know, and this aligns very well with our other gene editing projects, if you find efficient in vitro editors, you can actually find efficient in vivo editors. And with that, we can now move into our proof of concept studies and really ask, can we actually correct disease-related phenotypes? And we are using our lag dragger mice. They have a very severe survival defect, very severe hind limb paralysis. They are presenting us a very perfect model to get proof of concept studies. But then following that, and this aligns very similar with our existing gene editing projects, if things go the well we think and the way we hope they go, we can then actually use human cells to really screen and design human editors, making a step closer to the actual clinic. And sort of with that in mind, I want to thank you for your attention, but I also want to thank a number of people that are involved in these projects. Obviously, my boss, without her, none of this would be really possible. I like to thank Raj. He's working on a lot of the iPSC work in our group. There are a number of people in our group that I like to thank that you can see on the slide. Obviously, I like to thank Angus that he decided to take on this project. It always requires someone in David's lab. I also like to thank our funding sources. Without NIH, a lot of the work would not be possible. Thank you.
Any questions? I'll start with a question, Markus. So I think when we start talking about these kinds of gene editing therapies, we're starting with the leg dragger for the platform, but ultimately we would want to select more, especially that are represented in the patient community. How do we think about the number of patients or the severity of the mutation, and how did that get incorporated into the way that you guys set up your mouse and cell models?
I think in general, people are hoping that it is not going to require every single time a mouse. There are a lot of projects in the consortium that, very similar to KIF1 alpha, have a large pool of patient variants. And a lot of effort goes into the idea, and regulatory advisors seem to be very open about that you perhaps only require proof of concept for 1 or 2 animals. But ultimately, you might be able to rely on iPSC cells to then actually just develop the editor for any given patient variant, with the idea that it's the same principle. If we can correct one variant, if we can correct a second variant, why would we not be able to do that for a third, a fourth, and a fifth? I think that rationale makes a lot of sense, and I also don't think that it would be effective to create every single time a mouse just because there is one variant. I think when we just look into the room, if we would imagine how many animal models it would take, and if we also take into account that animal models not always work, this would be a huge amount of effort and perhaps money that could be put elsewhere. So I think a lot of regulatory people are realizing that we need for right now proof of concept, but in the long term, you probably don't need to do every single time a new mouse model.
We can take some online questions while Dylan goes to the next in room. The first question is, what do you predict the timeline would be from this to clinical trials and then to a rollout?
So for KIF1 alpha, I cannot really speak about a timeline. That's a little bit tricky because as we have learned, it is not just about the science. There is so much regulatory process involved, clinical aspects, physicians, which makes it very difficult for me as the, I look like 12, so it's like I can barely take any of this with me. But if we think about the SCGE consortium, which has a significantly bigger group of people behind it, a lot of the projects that are part of that are really working on a timeline of 3 to 4 years, which still sounds a lot, but is actually quite remarkable if we think about that. And I'm hoping, and this is similar to the consortium, that this will actually then allow us to figure out, or have figured out, how we can now do the other variants, how we can do the other genes. Because as Anne Loram has explained, they have a path. Gene editing doesn't have a path. Right now, researchers, FDA, as well as regulatory people, all have to figure out what should a path actually look like? What is efficacy that we want to see? What is safety that we want to see? Because we are manipulating DNA, so there is a reasonable risk and a reasonable concern about that. So it's really hard for me to give you an exact timeline, and whatever number I would come up with would probably be wrong.
Okay. Hi, I'm back here. Sorry. Great talk. I have kind of a question here, and I like the idea of editing. Obviously, this is a great idea. But can you consider a scenario, as with the ASOs, where we're targeting benign mutations, where editing could be used to introduce, say, a stop codon or some sort of targeting, so that we can, instead of doing 100 different mutations at a time, we can take the same approach and do that way. So, I mean, I like the idea, but how can we simplify this so that it's more clinically relevant?
So I think this depends on the disease. Yes, there are disorders where you can make it a more universally applied technology. I think SMA is a really good example. In this case, you actually only have one editor that can treat humans in general. But that is a really good advantage for SMA. When we think about KIF1 alpha, for instance, I don't think that you can use a benign SNP because you are correcting a specific nucleotide. You don't want to get rid of anything here. You actually want to reintroduce 2 fully functional copies, meaning it would be patient by patient. Now, that being said, when we think about other projects like baby KJ, which has a liver disease and very similar, it's very patient variant specific, people in the consortium, together with regulatory advisors, are considering sort of the idea, if you can find the prime editor and you can stick to the same prime editor and you only need to change the guide, you can make this a platform approach. You can get potentially through small-scale toxicity studies and then actually get it quickly being a turnaround, even if you have to make the guide variant to variant specific.
Yeah, totally understand. And there is a consortium, I think, right now in the NIH, right, or FDA,
Yes.
that is enabling that or empowering that idea and setting forth the guidelines for that. So great, thank you.
We have a couple questions online asking similar themed questions. Could you explain the difference between base editing and prime editing? And someone else asked, can you explain the difference between gene editing and something like CRISPR, or are they the same thing?
I would call gene editing and CRISPR the same. Like CRISPR is the Cas9 enzyme, so I think it's just the more commonly used name, like CRISPR. So those are technically the same. The difference between base and prime editing is base editing is a biochemical reaction that allows you to modify a single base and make it look like a different base. And this allows you to change the letter in the DNA from one letter to another. Prime editing is a little bit more universal. It allows you to change a lot more different variants than you can do with base editing. So base editing was a really great start, but now people are also exploring prime editing because it gives you a better window. It gives you more opportunity to correct different variants that you wouldn't be able to correct with base editing.
Thank you. And then one more from online before we turn back to the room. Why are you using viral vectors? Would something similar to administering systems like ASO work as well?
I think we are thinking about what can we do today and what can we do in 5 years. Viral delivery is right now the devil that we know. And in order to think about what can be done, in the next years, we use viral systems because we can deliver it more effectively. However, there are people working on alternative delivery systems. And I think when we think about labs like Ben Deverman, the Deverman lab, who recently developed new capsids, which are little shells that allow you to hide your little product that you want to get into the brain. Those are new technologies that potentially allow you to kind of go away from a more traditional viral approach. When we think about baby KJ, the people actually didn't use a viral system. They used lipid nanoparticles. Lipid nanoparticles work really great for the liver. People are trying to get those systems to work for the nervous system as well. So I think there's sort of a dual approach. What can we do today? And today is probably viral. But tomorrow there's probably something different, and people are working on what can you do tomorrow.
And the next online question is, can prime editing modify the entire motor domain of the KIF1A gene with one viral vector, or is there a limit to the number of edits?
There is a certain size range where you can edit within that range, so you wouldn't be able to modify the entire motor domain. The entire motor domain would be too big. But there might be approaches that you can target 1 or 2 or 3 variants that are close by at the same time. So you could have maybe an editor that would be applicable to a few people rather than truly just one individual. But in general, no, you wouldn't be able to modify the entire motor domain with the technology that we have today.
I'll just add to this that this is a really important thing for why we need additional engagement in our natural history study and more knowledge about where these mutations are, because the more information we get from families, the more crowded the mutations become on our graph. And so knowing where those clusters are in the relevance of that size limit could actually be really important when we're talking about what we do once this platform is set up. So just another call to action that the more info we have, the more info we can pass on, the more informed our therapies can be. So something to keep in mind. All right. I saw one question about how families can help.
You are not a paid actor. As a parent of KIF1A, how can I help accelerate your mission to reach clinical trials? Is technology, regulatory, or funding the main issue?
I think it's all of the above. I think when we look at it, it's all of the above. I think it often starts for us with research, which is, the work in the lab. But I think spreading the word, having, conversations with whoever you can reach out from a regulatory perspective is important. I don't think that one is more important than the other. I think it's always a preparation of the next step. Am I going to be unhappy if someone wants to support my work? Absolutely not. But I also know that this, people have to put the money sometimes they have to put the money somewhere else in order to make things work. And we usually try as much as we can to use public funding to support our work. So if foundations, parents can put their effort somewhere else, we still appreciate that because it's about spreading the work, getting partners together.
Hello. Thank you so much for this. A question I have is, so I'm in the DC area, and friend of a friend is someone that I think knows you well. Her name
Does this person start with Nina?
Yes.
Nina told me.
I'm very happy to meet you. But in talking to her, so her daughter also has a rare disease, and she helped start this amazing organization for AHC. I guess I'm just wondering, how can we collaborate in a meaningful way, or KIF1A, the organization itself, just with other organizations on a smaller level or medium level, bigger level?
I usually like to have access to the patient foundation, and I think when we consider talks like from Wendy yesterday, having access to natural history data is incredibly helpful, especially when we try to understand mouse models. It usually helps to have a sense about what do people see in the human population. And therefore, as much as people can give information to Dylan and Dominique, please keep doing that, and then I can bother them to get information. Equally, all the genetic information is helpful to understand what are the variants that are there. And I think in general, it's really just spreading the word, like the way Nina does it. Nina gives us input, and I think this is equally, how Patient Foundation do it in general. What are outcome measurements that are important to you? And that's what we did with Nina. We were asking Nina, where do you want the therapy to have, a correction in the context of an animal?
Where that organization is in what they're doing right now, is it I feel like I don't understand actually, but like what they're doing versus what we're doing, is it in a similar place or?
It is in a similar place. It's just like, every patient foundation is doing slightly different. I think if you wanted to say how different or similar it is, imagine Nina to be Dylan.
Okay, gotcha. Cool.
Like in one sentence, Nina is Dylan.
Okay, that's very helpful.
Or Dominique. Like, I don't think that you can separate them necessarily, but yes, this is how I would describe Nina.
Gotcha, thank you.
I'll be going by Nina for the rest of the day. Let's give a big round of applause for Markus.